mic is yours. Thank you, everyone. It's going to be a great day.
Developing, so at the green fruit stage, you can still see see at first. At the green fruit stage, the fungus colonizes from inside the fruit. You can't see it just yet on the surface. But later at the fruit ripening stage, the fruit turns pink, segments, and turns white in the end, drops off the plants. But here you see some photos of different stages of fruit ripening, and on the right you see that the fruit is cut open, and if the fungus was colonizing from within. And you can see in some scenarios, such as this middle one here, is you don't see it on the outside, but from the inside the fungus is already colonizing the tissue. Oops. Here's an example of how the fruit symptoms look like at a later stage. So here you see the turning pink, and they shrivel, turn white, and that's how they appear at the plants at the final stage in the season when most of the fruit is already harvested. The overwintering of this fungal pathogen occurs in the infected berries. So this is the end stage of how it was on the plant. Then it drops off the plant on the soil surface, and it looks like this. So they're still white, but they're starting to darken, and they have this pumpkin-shaped structure. And this is how they appear when they're in the middle of winter in their dormant state. So just as the plants go in a dormant state, this fungus also goes in a dormant state. But they're particularly hard to find in a soil surface when they appear as such. And in spring, when under the warmer environmental conditions, these mushrooms or apothecia appear from these dormant structures, as you can see in this photo. So you see these little brown mushrooms appear from the berries. They're very small, as you can see in this photo. And they have several stages of development. So here from left to right, you see the different stages that Flores and Pshai at Oregon State University have distinguished and are officially used in the terminology now. So this is the dormant stage. Then we have the second stage when the first initials show. We call that germination. The emergence of the apothecium, these fleshy structures, we call emergence. Then when the cup starts to appear and you get the indent in the tip, it's called differentiation. And when the open cups are producing spores, we call it sporulation. Now what this ascospore release looks like, so ascospores are the spores that are released by these apothecia. I have a small video. It's made in Michigan State. So you see it's under wind and environmental conditions. I think in this video, they released the lid from the container that they were keeping these apothecia, and that makes them to release all the spores at once.
That's not time lapse, that's real time.
Sorry?
That's actually what it looks like when it happens, it's not a time lapse.
It would happen as such, but you wouldn't see it as apparent. Like if you would actually look at it, you wouldn't see it as a puff like that. Yeah, it's more of a staggered response. Oh. Okay, so this disease causes up to 70%, or I've even heard 85% in individually affected fields, and it's particularly hard to manage once it is established in your field. Current control suggestions are based on the calendar basis with routine spraying of fungicides, but not based on the fungal biology. And most of the research is performed in eastern USA, with the blueberry growing regions there, which have very different climates. And so the recommended strategies they have there, or the research that they did there, might not be applicable to this area. So therefore, we started investigating what the disease cycle looks like in this region. And so here I'm going to go through the disease cycle of this disease. So as you saw in the photos before, in the winter we have this dormant structure, and in spring the apothecia develop and produce ascospores. The ascospores are disseminated by wind and infect the emerging flower and leaf tissues from the buds, and secondary, it creates these flower and shoot strikes, and on the surface of these samples is a secondary type spores, which is different from the first phase, being conidia. And this fungus is very unique in that it mimics the pollination process. So it secretes a sweet odor and reflects UV in such a way that it attracts insects. So the insects land on the surface of these symptoms and attach the conidia on their furs. And then they take this fungus into the flowers during their pollination process. So these spores land on the stigma of the flower and colonize the stylar canal into the ovaries of the flower, and then start colonizing the fruit from the inside. So that's why you don't see it in the green fruit stage, but then once the fruits start developing, it starts turning pink, the more it gets colonized by the fungus. And eventually it ends up in the dormant state on the soil surface again. Now, in order to protect the crop from this disease to occur and the spores to reach the crop, it's important to protect, to time the management and protection regulations coinciding with this ascospore release to prevent the spores to get to the susceptible tissues. And that's why I focus my research on this stage of the disease cycle. Oops. Oh, now here is an example of a management, recommended management from the Eastern United States. So this is a slide by a professor in New Jersey. And I just like to go through it a little bit. So here on the top, you see the different fungicide groups that are currently used for management. So for those two phases of the disease cycle, you see that there's different type fungicides recommended. So for the primary phase, what is suggested now is to look at the vegetative bud development, and when the first green tissue is developing, here, like here, is when it's recommended to start applying these fungicides. And then once the floral period starts for the crop, there's these different fungicides that are applied that are also covering the other diseases such as alternaria and anthracnose. Anyway, we're gonna come back to this slide at a later stage. So the objectives of my research are to determine the timing that this spore release is occurring in this area, to determine environmental factors that are influencing this spore release, to determine differences in timing of bud break of cultivars grown in this area, as in the timing that these emerging tissues are appearing on the crop might be different between cultivars depending on their early maturing, late maturing. So I wanted to investigate if in commonly grown cultivars in this area, there is a timing difference, and therefore the timing difference may make it more susceptible by coinciding with this spore release period or not coinciding. And the fourth objective was to develop a prediction model. So in this research, I studied the 3 factors that are involved in calculating this infection risk. It's the pathogen, the host, and the environment. And I'm starting with the first one, the pathogen. So in order to determine when this spore release is occurring, in both the 2015 and 2016 seasons, I scouted for these epithelial abscess presence in 3 fields, which were 2 fields in Washington, one in Whatcom County, one in Skagit County, and one in Benton County in Oregon, where Professor Jabe Scheid provided the data. I used Burkhardt spore traps, which are depicted here on the left, to capture ascospores out of the air and determine if ascospore release is agreeing with my observations of apothecia or no apothecia. Here you see the results of this study. So you see in the bars are the different locations and the different patterns are different years. So in green and blue, you see the results for the Washington fields. Ascospore release was observed from beginning of March to mid-April. And this is a very long period. It was 37 days, and this is longer than other periods that have been reported. On average, it was about 16 days in the eastern United States. And therefore, it shows that this in Washington is indeed very conducive for this pathogen and producing apothecia. In Oregon, the timing was slightly different, so it started later, and between the 2 years, there was a big difference in this period. And this could potentially be caused by the different soil types between the 2 states that might be drying out or staying wet at different rates, influencing the environment that the apothecia need to thrive. So that leads me to investigate the environment. So in order to determine what environmental factors, out of all these factors that we can measure these days, do influence these apothecia, I used 2 systems, the WSU Ag WeatherNet and in-field weather stations, to see if if you're not familiar with WSU Ag WeatherNet, it's a publicly accessible network of weather stations, whereas in-field weather stations are sensors that can be placed in your own field or preferred spot. I wanted to see if the WSU Ag WeatherNet can be used by everybody to predict their infection risk of these ascospores, just to see if you can use that or that you need to have purchased your own in-field weather station to calculate it appropriately. Now, the factors that I included in both systems were air temperature, relative humidity, leaf wetness, and soil temperature. Precipitation and solar radiation of WSU Ag WeatherNet were included, and soil moisture sensors were placed in the field. And now, I did several analyses and modeling approaches, but the 3 factors that came out as the most significant were soil temperature, soil moisture, and solar radiation, being light. And to get an idea of what the level of these, or the thresholds of these factors are, these are presented here. So here are the daily averages for a month before. This is the average of the period that apothecia were found, and this is a month after. So you can see the soil temperature of about 50 Fahrenheit, 43% of soil moisture, and 12 megajoules per square meter were the average most conducive environmental factors. And in these analyses, we also found that the WSAC WeatherNet data can be used. However, WSAC WeatherNet doesn't have soil moisture, and this was one of the most significant factors. So there would still be a need to place either a sensor or other way to measure soil moisture in the location that you're interested in. Moving on to the third factor, being the host. So from literature, we know that the blueberries are only susceptible at very particular stages of the floral and vegetative bud development. And these are bud burst and tight cluster stages for flowers, and late green tip. So when more than 1/4 inch of green tissue is available from these buds. And what I was curious about is the timing that these stages are appearing on the plants. So in Waukesha County, I monitored Blue Crop, Duke, Liberty, and Draper in 3 fields for each cultivar per county. I took photos of the shoots every week and determined the stages from photographs. And here you just see the results of this experiment. So these are the graphs representing the amount of susceptible floral buds out of the total floral buds measured. And the period of this graph presented is the Ascospore release period. Now, in the different colors, you have the different cultivars, and in the continuous or dotted lines are the different counties. And well, what I like to point out is that you can see that the green and blue lines, being Blue Crop and Duke, had their peak numbers of floral susceptible buds available earlier than Liberty and Draper. And this was observed in both years. And also, you can see that the high numbers of susceptible floral buds were appearing early in the Ascospore release period. Now moving over to vegetative buds. So these graphs depict the same story as I just presented for flowers, but for vegetative buds. And you can see here that the blue, so being Blue Crop, had its peak number of susceptible leaf buds earlier compared to the other cultivars that showed a lot of variation. But you can see also in 2016, it was already coming down from having these peak numbers available, and therefore potentially avoiding this Ascospore release period and being less susceptible. Now in these graphs, you see a comparison of the different cultivars and the amount of susceptible tissue they had on the plants during this Ascospore release period. So it's a summary of the amount of tissue during that period. And the different colors are the different cultivars. On the left, you see the graphs for floral buds, on the right for vegetative buds. And overall, you can see a big distinction that there was way higher amounts of susceptible floral buds compared to vegetative buds, which is very important. And then between the cultivars, we saw that Draper and Liberty had a higher number of floral buds and vegetative buds. Blue Crop had the highest number in 2015, but Liberty had the highest number in 2016. So from this results altogether, it shows that Liberty, for this particular Ascospore release period, was the most susceptible, merely based on phenology, but based on developmental stages that were presented. Now, just summarizing what we found so far and how the information can be applied in the field. So regarding the timing of Ascospore release, we found that it's a very long period compared to other areas, and it was found between the period of early March, mid-April. And the application of this information is that the plants will also need protection for this long amount of period. The second question regarding the environmental influences on apothecial development, we found that soil moisture, soil temperature, radiation had a significant effect, and therefore should be monitored for estimating the infection risk and timing that Ascospore release is occurring. And then the WSAC WeatherNet can be used or in field weather stations. However, the soil moisture is an important factor that should be included. Moving to the findings in regarding the host, we found that floral buds show their susceptible peak numbers of susceptible buds earlier and in higher numbers compared to vegetative buds. Now, remember I presented this slide about this New Jersey management program where they only monitor the vegetative buds. And so this research shows that it's important to also monitor the floral buds, as if you're missing that, seeing that they're earlier, you might also miss the timing that the first application should be applied. Well, also we found between the four cultivars that were included in this study that Blue Crop and Duke had higher numbers of susceptible tissue earlier in the season compared to Liberty and Draper. So depending on it coinciding with Ascospore release, these cultivars may be more or less susceptible. In this case, Liberty and Draper were more susceptible. Now, this is the same slide as I presented before, but what I would like to correct there is that now we need to make sure that we don't only monitor the vegetative buds, we should also include monitoring of the floral buds for timely application of management practices. Now, this last objective of my research was to determine or to develop a prediction model for the infection risk or the Ascospore release. And so the information we found so far that can be used for prediction are, well, you need to know if you have a history of mummyberry in your own field or in neighboring fields. The second factor would be the environmental factors. So to monitor those three factors and see when they reach these thresholds, and to the susceptible host stages. So the infection can only happen when these three factors are present. Now, to make this a little easier and provide a product that can help us predict this Ascospore release, I worked with Dr. Michael Grant at University of Washington, and we've been working on an application to do this, and I'm going to try and show you what this looks like. So here, this would be the screen you see either on your phone or your desktop. And then you can insert values for the three weather factors, so soil temperature, soil moisture, solar radiation here, and the stage that your flowers or leaf buds are at, and you press predict. And then you can get this donut that shows the percentage risk that you're at based on the values that you entered. And so when you change them, here if these are closed, the risk changes. So this is what we've been working on. It's still under development, but I really wanted to show it to you today. And hopefully this will be a product that can be placed on the website or as part of the Wacom newsletter or extension, one of the extension web pages. Okay. Now, for validation of this, so yeah, in this application that we were wanting to put online, we use an algorithm, and to see if this algorithm actually predicts the apothecia accurately, this year I'm validating this model with 4 field locations in Washington, so 1 in each of these counties, and optimizing, validating this model this way before we make it, we release it officially. And to just get an idea of what the current status is of apothecial development in the area. So here's these fields in the different locations. And on the bottom, you see the different developmental stages of apothecia. And so it looks like overall it's similar in each of these counties. However, in Whatcom County, it's a little more progressed, where more apothecia, well, 12% are at the differentiation stage. So they're not producing the spores yet, but they're starting to get to that stage just before where the cups are not opening yet. So yeah. Oh, and of course, the mummyberry updates are getting circulated again. So these mummyberry updates present the stages of flowers and leaf buds, as well as the apothecia in Whatcom and Skagit County, to get an indication or help the community to estimate when the disease management applications or strategies should be applied. And so with that, I'd like to thank well, a lot of the cooperators that we've worked with today, firstly. But then funding was provided from the Washington Blueberry Commission, Washington State Commission for Pesticide Registration, and the Northwest Agriculture Research Foundation to do this research. So, and then again, I'd like to thank the blueberry growers, the small fruit pathology team, Drape Scheid of Oregon State University, and Michael Grant of University of Washington for helping me program, and the Northwest Research and Extension Center. So thank you.
Questions for Dumpy? Craig?
Yeah.
Is there any indication of how far those windborne ascospores will travel after termination of the
Yeah, so the question is, do I know how far the ascospores will travel after they're disseminating? There's some conflicting reports. I've read 30 meters to 300 meters, which is, well, how many feet is that? To 1,000 feet, yeah. So that's the only answer right now.
Any other questions? Jules?
How do you sign up for the mummyberry report?
The question is, how do you sign up for the mummyberry report? So the mummyberry report can be found on our website, but it's also circulated by the Washington Blueberry Commission as a PDF every week. And it's been posted on several extension websites, like on Facebook. And it was first posted at the newsletter as well. Yeah.
All right. There's no other questions. We'll move on to our next speaker. Let's give Dumpy a big round of applause. All right. Next up, we have Dr. Tobin Peaver.
Need to take computers, change it.
Change computers. So we're going to take just a 1 minute break to swap out some computers, and we'll be right back.
Okay.
Thanks.
Suppose you can adjust it. Holy cow, you're really hooked up.
Yes, I am. Oh, yeah.
Wow.
How does that feel?
I don't think I've ever been this wired up in my life. So can these be together, or does it matter?
Yeah.
Okay.
It should be at the same place.
Just so you're aware, the down arrow advances the slide. It's
Okay.
really counterintuitive.
I know. It's all I've always some of these, you know, they got them side by side, and you're always doing the wrong. So which one's the pointer? This one?
No, it's in the middle.
Pointer's in the middle. Okay. That's advanced.
Yeah.
Okay. This should be all I need to know. So can you get my? So what are we have 40 minutes technically between each one?
I think they're all different, so
Okay.
I'll look at it.
Just let me know if I'm, because I got some extra slides if I have extra time. Oh yeah. Test. Okay. Can you hear me? Test. It's good? Okay. Oh, it should be on the desktop, I think. Oh, I don't know with the
Hey,
with the
Chris.
meeting thing. Hey, Chris, I think we need your help here.
You're all good.
Just minimize that.
Okay, so we can minimize that. Don't.
Don't push that button. Oh, something. Might be. Hold on, no. Should be
Everybody's got that.
that one.
Let's find out. Author DOC.
No, that's Hartville. That one.
Can you just make sure it's on presenter
Because that looks like it's the older version or something from the. Nope, that's Alan Schreiber. I thought I put it on the desktop.